Since we have focused on the way that smooth muscle within the blood vessels of the systemic circuit, since we focused on how that smooth muscle contracts, it seems fitting to talk about how that same smooth muscle relaxes. Now, for this conversation, we really mostly are talking about the smooth muscle that exists within the small arteries called arterioles. How does that smooth muscle within our small arteries that exist within the systemic circuit, how does that smooth muscle relax? There's many, many different ways that it does, but one of the most known ways that it does relax is through nitric oxide. So we're going to take a look at how smooth muscle within systemic arterioles relaxes through the nitric oxide pathway. To understand how nitric oxide causes smooth muscle within blood vessels to relax, you first have to understand the very basic anatomy of blood vessels. So both arteries and veins generally have three layers. We're focused on arteries, but this is true for veins too. There's the innermost layer called the tunica intima. Tunica intima largely is made up of epithelial cells. These are called endothelial cells, and they are what actually face and make contact with the blood that's traveling through the lumen of the vessel. Then the artery has a middle layer, which is where all the smooth muscle is that we've been talking about, the tunica media. And then there is the outer layer, which is a layer of connective tissue, which is called the tunica externa. What's important is to recognize where these different layers are in relationship to the blood that's flowing through the vessel. What this image is depicting is blood traveling through a blood vessel. Now, I've already said this, and that was the reason I briefly mentioned the anatomy in the last slide. As blood travels through this blood vessel, what does it make contact with? Blood makes contact with the innermost layer of the blood vessel, which I'm highlighting in black there. That is the tunica intima. It's made up of epithelial cells. It's called the endothelium. And as the blood flows past this tunica intima, these epithelial cells, it creates something that's known as shear stress. Shear stress is basically a frictional force that is exerted onto the epithelial cells that exist within the tunica intima, again, collectively known as the endothelium. And this shear stress can be less or it can be greater. For example, if somebody's involved in a lot of physical activity, exercise, shear stress tends to increase. Basically, more blood is traveling through the vessel. The vessel shape could be potentially becoming deformed for a variety of different reasons due to the exercise. There's many different things that could influence this, but generally speaking, yet again, high intensity exercise, if the velocity of blood is traveling through the vessel, if it's increasing, if there's more blood within the vessel, which can happen certainly as well and does happen, both of those things are going to increase the frictional force that the blood creates against the tunica intima, against these epithelial cells. So what we would say is that, again, during something like exercise, shear stress within blood vessels, specifically systemic arteries, and even better yet, systemic arterioles, that shear stress can increase pretty dramatically. And what can that do exactly? And another way of saying this is that during something like exercise, the blood flow through these vessels, more specifically the systemic arterioles, that blood flow starts to become much more turbulent.
All right, you have to pretend that this is an epithelial cell called an endothelial cell lining the arterial wall. I'm drawing a bunch of them for you. What's traveling through this up here, this is all And here is the lumen that blood is traveling through. Okay, blood's traveling through the lumen of the arteriole. Now, this arteriole during exercise, right, is getting squeezed, it's getting manipulated, and lots of shear stress is occurring, turbulent blood flow through this vessel. Okay, and that turbulent blood flow is felt by the epithelial cells that line the vessel, that line the arteriole. Okay? So what this is showing is shear stress, and the shear stress is, let me erase this. The shear stress is mostly being felt and experienced by the epithelial cells. Okay, so that turbulent blood flow is happening and being exerted onto the epithelial cells, the endothelial cells lining the arteriole. And when that shear stress is experienced by the epithelial cells, we think that it activates a pathway. It might do a couple things. Ultimately, it's going to allow for calcium levels to elevate inside of the endothelial cell. But how that happens is, well, somewhat hard to know, but we have a good idea of the pathways. Okay, so one possibility is that the shear stress that's experienced by the endothelial cell, or what that does is the shear stress itself opens up calcium channels within the plasma membrane of the endothelial cell. And that allows calcium to spill in to the endothelial cell. Other possibility is that shear stress actually activates a G protein pathway. And this is a PIP2-IP3 pathway. Okay, so shear stress occurs, and that activates actually a G protein pathway. The alpha subunit associates away from the beta and the gamma, activates phospholipase C. Phospholipase C converts PIP2 into IP3 and DAG, and here you're seeing IP3 levels increasing inside of the endothelial cell because of the shear stress it's experiencing. And what IP3 does is it then causes calcium to be released from the endoplasmic reticulum of the endothelial cell. So no matter which one of these happens, you're getting an increase in intracellular calcium levels within the endothelial cell because of the shear stress. Either the shear stress on the endothelial cell is directly opening calcium channels, or it's activating an IP3 G protein pathway that's causing calcium to be released from the endoplasmic reticulum. Moving on, what does calcium levels elevating inside of the endothelial cell do? Well, calcium can combine with calmodulin inside of the endothelial cell. When calcium combines with calmodulin, in this case, it doesn't activate a calcium calmodulin dependent kinase or myosin light chain kinase. It does nothing like that. Instead, what it does is it binds to what's called nitric oxide synthase, eNOS. You can see there. That's nitric oxide synthase. And calcium calmodulin binds to and activates nitric oxide synthase, and nitric oxide synthase begins to create nitric oxide, or NO, within the endothelial cell. So what does shear stress do to the endothelial cells? It causes them to start producing nitric oxide. And it does it, we think, through elevating intracellular calcium levels within the endothelial cell. So now the endothelial cell is experiencing shear stress during exercise and is producing all this nitric oxide. And what is the nitric oxide? Where's it going to go? What's it going to do? All right, so you're getting very turbulent blood flow through this arteriole. Okay, it's being squeezed by contracting muscle fibers and other things going on, and that's causing the blood flow through it to be very turbulent. And it's then causing these endothelial cells that make up the tunica intima to produce nitric oxide. Right? They are, the shear stress is probably causing them to release calcium, binding to calmodulin, activating nitric oxide synthase. Okay, so they're producing nitric oxide. What the nitric oxide does is it spills out, and then it enters into the smooth muscle cells that line the tunica media. And now what's happening is the nitric oxide is within the smooth muscle cells in the tunica media. And we want to see, what does nitric oxide do to these smooth muscle cells? Here is nitric oxide entering into a smooth muscle cell. When nitric oxide enters into a smooth muscle cell, what it does is it doesn't activate adenylate cyclase. It activates something that's relatively similar called guanylate cyclase, or guanylyl cyclase. Depending upon the pronunciation, you can also call adenylate cyclase adenyl cyclase. So we'll call this guanylate cyclase. Guanylate cyclase is activated by nitric oxide, very similar to adenylate cyclase. But here's the thing. Guanylate cyclase is different than adenylate cyclase because it converts GTP to cyclic GMP rather than ATP to cyclic AMP like adenylate cyclase does. So these are very similar but slightly different. Nitric oxide activates guanylate cyclase, which starts converting GTP, which is a lot like ATP, into cyclic GMP, which is a lot like cyclic AMP. What cyclic GMP does is it activates what's known as PKG. PKG is actually what's called cyclic GMP dependent kinase. It's a lot like PKA. You know how cyclic AMP activates PKA? This is very similar to that, except this isn't PKA. It's cyclic GMP dependent kinase. That's what PKG is. So cyclic GMP activates PKG rather than PKA. But cyclic GMP dependent kinase, is a kinase. It's going to go around and it's going to start phosphorylating things. So the point is, nitric oxide enters into the smooth muscle cell. And if you follow it downstream, you're going to get an increase in PKG within the smooth muscle cell. Now, PKG can do a lot of different things, we think, and some of them are probably true, others are not. We don't fully understand. But one of the things that PKG can do is it can phosphorylate and knock out myosin light chain kinase, we think. Okay, so if PKG phosphorylates, it's a kinase, so it phosphorylates things. If it phosphorylates and knocks out myosin light chain kinase, then there's going to be nothing to phosphorylate the light chains on the myosin heads, and thus the smooth muscle cell will not be able to contract. This is similar but a different pathway to ADP and adenosine,
which are other metabolites that are believed to contribute to muscle relaxation, especially during exercise. But going backwards, I mentioned a little while ago that if myosin light chain kinase can get phosphorylated by something, that will essentially knock it out and prevent the smooth muscle from being able to contract. And we think that's more or less exactly what nitric oxide does to smooth muscle cells that exist within blood vessels, particularly smooth muscle cells that exist within arterioles.
Additionally, it's possible that PKG is able to activate the SERCA pumps on the sarcoplasmic reticulum within the smooth muscle cell. And if the SERCA pumps are active to a greater degree, what's that mean? They're going to pump a whole bunch of calcium back into the sarcoplasmic reticulum, and then there's not going to be calcium to bind with calmodulin, and there's going to be less myosin light chain kinase, and thus there's going to be less phosphorylation of the myosin heads. Basically, myosin light chain phosphatase will be more active than myosin light chain kinase, and you'll get relaxation. So in this theory, kind of PKG is able to remove intracellular calcium by activating the SERCA pumps to a greater degree, and thus causing relaxation of the smooth muscle cell.
So as we've been talking about smooth muscle contracting within systemic arteries, the pathway that we looked at for that to happen is through activation of adrenergic receptors. Norepinephrine binding to them, which activates the pathway, which allows for the smooth muscle to contract. Another pathway that can allow for smooth muscle to contract, to a lesser degree within blood vessels and to a greater degree within other organs throughout the body, is if the smooth muscle cell depolarizes, if the interior becomes more positive, That depolarization will cause voltage-gated calcium channels in the plasma membrane of these smooth muscle cells to open. And that will allow calcium to spill in, and that influx of calcium will cause the smooth muscle cell to contract. I'm not going to go through the whole pathway by which this happens, because there's a variety of things that would happen afterwards to contribute to how this would cause the contraction, and I'm not looking to go through that whole pathway. I just wanted to mention this because there is another possible way that nitric oxide causes smooth muscle cells to relax. And it's through PKG targeting the availability of these voltage-gated calcium channels to open. So,
additionally, PKG may be able to open up potassium channels within the smooth muscle cell. That would allow potassium out, which would cause the interior to become more negative because it would be losing positively charged potassium ions. The interior would hyperpolarize, and the voltage-gated calcium channels within the plasma membrane would not be able to open up, and calcium would not be able to come in through that mechanism. That would also contribute to relaxation of the smooth muscle cell.
And this mechanism would probably be more relevant for smooth muscle that exists in other organs and less so within blood vessels. But nonetheless, this is another means by which nitric oxide may be able to cause smooth muscle to relax.
What can we collectively say about nitric oxide? Nitric oxide causes smooth muscle cells to relax. How does it do that? Nitric oxide increases PKG, the activity of PKG, and then PKG might do a variety of different things. It might knock out myosin light chain kinase by phosphorylating it directly. It might knock out myosin light chain kinase indirectly by activating the activity of the SERCA pumps, or increasing the activity of the SERCA pumps and reducing intracellular calcium. Or it might hyperpolarize the smooth muscle cell by opening up potassium channels and preventing calcium from coming in from the voltage-gated calcium channels. All of these things make nitric oxide a potent vasodilator. Okay? It will cause the smooth muscle within those particular arterioles to relax, and the arterioles will dilate, and you'll get a reduction in resistance and very good blood flow again through these arterioles.
Point being that shear stress is largely a good thing. It can cause endothelial cells, epithelial cells that line the lumen of the blood vessels to produce nitric oxide, which then causes smooth muscle to relax, and that causes the blood vessels to dilate, which of course can reduce blood pressure, which we know is very important for improving human health. And I want to point out here that all we did was scratch the surface as to what can cause smooth muscle to contract within blood vessels and what can cause smooth muscle to relax within blood vessels. We only talked about smooth muscle contraction through activation of the sympathetic nervous system releasing norepinephrine onto those blood vessels. But there are different receptors on different blood vessels where the same norepinephrine can stimulate contraction of that smooth muscle through different pathways. And vasodilation can happen within smooth muscle that exists within blood vessels through a variety of different neurotransmitters that are released onto those smooth muscle cells. You can see many of those different mechanisms there. Additionally, there are different hormones and other chemical messengers that can stimulate vasoconstriction, and we didn't talk about any of those. And there's much more than just nitric oxide that can cause vasodilation of smooth muscle. So this could be literally an entire semester-long course, almost just going through all of the different means by which smooth muscle within blood vessel can be caused to contract and relax through different neurotransmitters and through different hormones and chemical messengers. And mind you, this isn't even talking about the smooth muscle that exists within other organs. So the control of smooth muscle is wildly complex, and that is why entire textbooks are written about it. But hopefully this gave you a nice little introduction to some degree as to how smooth muscle is organized structurally and how it functions in different circumstances.